Close your eyes for a second. Imagine the frozen tundra of the Ice Age. A shadow emerges from behind a snowdrift, and you see it. A colossal saber-toothed cat with dagger-like fangs. Sounds like science fiction, right? What if I told you that the brightest minds on the planet are actively working to turn that fantasy into reality? This is Project Resurrection, and its goal is to bring extinct giants back to life. In this video, we'll break down every detail, from hunting for the perfect ancient DNA to navigating the incredible ethical dilemmas. By the end, you'll know whether we might actually see a living Smilodon someday, or if this will forever remain just a dream. By the way, do you think it's even worth bringing back apex predators from extinction? Drop your opinion in the comments right now. Let's start a discussion. Before we dive into the world of genetic engineering, let's meet our main character. The saber-toothed cat, or more accurately, the Smilodon, wasn't a true tiger in the modern sense. It belonged to an entirely different genus of felines that roamed North and South America during the Pleistocene epoch. It vanished about 10,000 years ago, alongside the mammoths and the giant ground sloths. Its most defining feature, of course, were those incredible 7-inch canines. But this was no mere cat with big teeth. The Smilodon was a powerful, stocky predator, built like a cross between a lion and a bear, perfectly engineered for hunting megafauna. The idea of bringing it back to life is part of a much broader concept known as de-extinction, or species revival. The most famous candidate for revival is the woolly mammoth, but the saber-toothed cat is the next hurdle, a far more complex and intriguing challenge for science. So, where does any resurrection project begin? Naturally, with the hunt for genetic material. The first and most crucial step is locating a sample of saber-toothed tiger DNA that is sufficiently well preserved. The ideal place for these finds is permafrost. Tissues frozen solid in the ice can preserve biological material for tens of thousands of years. That's precisely why scientists have such a wealth of material on mammoths, thanks to discoveries in Siberia. But there's a significant problem when it comes to the Smilodon. They lived in warmer climates, primarily across what is now the United States. You don't find those vast stretches of permafrost there. The primary finds come from bones and teeth discovered in the La Brea tar pits in Los Angeles. While the asphalt is excellent at preserving bones, it is devastating to fragile DNA molecules. Therefore, searching for an intact Smilodon genome is like looking for a needle in a haystack that, incidentally, has already been set on fire. Even if scientists manage to strike gold and find a viable specimen buried deep in a frozen corner of Alaska, they immediately face the next hurdle: DNA degradation. Time is brutal on genetic material. A DNA molecule has a known half-life, estimated at around 521 years. This means that 10,000 years after the last Smilodon died, its entire genome would be reduced to nothing more than tiny, scattered fragments. Imagine taking the novel War and Peace, running the entire book through a cross-cut shredder, and then attempting to reconstruct it. You wouldn't have whole pages, just microscopic scraps of words and letters. Trying to restore a coherent, meaningful text from that mess is a virtually impossible task. That's precisely why the idea of cloning dinosaurs, whose DNA is millions of years old, remains pure science fiction. While the saber-toothed cat offers slightly better odds, the problem of fragmentation remains the single biggest headache for geneticists today. But how exactly do scientists plan to tackle this billion-piece puzzle? This is where supercomputers and bioinformatics come into play. The process is called genome sequencing. Specialized machines read every surviving DNA fragment, and then powerful algorithms attempt to piece them together in the correct sequence, just like a giant jigsaw puzzle. To figure out how to assemble this, scientists need a template, what's known as a reference genome. This is the complete genetic map of the closest living relative. In the case of the woolly mammoth, that relative is the Asian elephant. But who is the closest relative of the Smilodon? Surprisingly, research has shown that modern tigers and lions are not its closest kin. They diverged on the evolutionary tree more than 20 million years ago. This presents a massive complication. The more distant the relative, the harder it is to fill in the gaps in the ancient DNA, simply because there will be too many genetic differences. Let's imagine that after extensive research, scientists finally agreed that the African lion is the best available candidate for the template role. Their genomes are more than 90% identical, but those remaining few percentage points, that's everything that made the Smilodon what it was, its sheer size, raw power, gigantic fangs, and perhaps even its striped or spotted coat. Now for the most exciting part, genetic editing. This is where the revolutionary CRISPR-Cas9 technology comes into play. In simple terms, these are molecular scissors that allow scientists to cut segments out of one species' genome and replace them with genes from another, all with incredible precision. Scientists will have to take the lion's genome and make thousands, if not tens of thousands, of targeted modifications to transform it into the full saber-toothed tiger genome. This is painstaking labour, comparable to a master restorer meticulously reconstructing a lost masterpiece piece by precious piece. The process of editing the lion's genome would unfold like this. A team of geneticists starts by taking the complete DNA of the African lion and comparing it against the reconstructed, yet incomplete, Smilodon genome. Specialised software would highlight all the discrepancies. Then, utilising CRISPR technology, scientists would begin the methodical process of tweaking the lion DNA. For instance, they would locate the gene responsible for the lion's canine length and replace it with the Smilodon version, which codes for the growth of those giant, saber-like fangs. This same procedure would apply to genes governing muscle mass, skeletal structure, and perhaps even coat coloration. It's important to understand this isn't about creating a new organism from scratch, but rather a profound modification of an already existing one. Every single replacement is an incredibly complex operation at the molecular level, and thousands of these delicate procedures would need to be carried out until the resulting lion cell becomes genetically indistinguishable from the cell of the extinct predator. So, after months, or perhaps even years, of painstaking lab work, scientists finally have a complete, edited Smilodon genome. But right now, it's nothing more than raw data, a digital code. To turn this code into a living organism, they must introduce it into a cell and trigger cell division. This is where a classic technique comes into play, somatic cell nuclear transfer, or SCNT. This is the exact process used to clone the famous sheep, Dolly. Scientists will start by taking an egg cell from a female African lion. They'll remove the egg's original nucleus, which contains the lion DNA, and replace it with a new nucleus holding the synthesized Smilodon genome. Next, they use a mild electrical pulse to stimulate the egg. This essentially tricks the cell into believing it has been fertilized, prompting it to begin dividing. If all goes according to plan, within a few days, they will have a microscopic saber-toothed tiger embryo ready for the final and riskiest stage. Science now faces perhaps the biggest hurdle in the entire project, finding a suitable surrogate mother. The Smilodon embryo requires 90 days of gestation, and the African lioness is the only viable candidate for the job. This is where the real problems begin. The mother's immune system may recognize the embryo as a foreign body and initiate rejection. Even among closely related species, minute biochemical discrepancies can trigger a miscarriage. Furthermore, we still don't fully grasp the delicate biological choreography required between the embryo and the maternal organism. Moreover, pregnancy is a complex hormonal dance. The crucial hormones produced by the developing Smilodon fetus might not align with what the lioness's body is designed to expect, potentially leading to severe developmental failure. This hormonal mismatch is one of the primary reasons why successful interspecies gestation is exceedingly rare, even among modern existing species. Beyond biochemical incompatibility, there are purely physical challenges to consider. Although related to the modern lion, the Smilodon had a dramatically different build. It was shorter, but much more massive and stocky, possessing thicker bones and incredibly well-developed musculature, especially around the shoulders. An adult Smilodon weighed up to 300 kilograms, while a large lioness typically weighs around 180. This implies that even a Smilodon cub would likely be significantly larger and heavier than a typical lion cub. This places an extreme additional strain on the surrogate lioness during the pregnancy. Delivery could prove extremely complicated, or perhaps even impossible without a caesarean section. Any deviation in fetal size or growth rate could easily become fatal for both the cub and the mother. Science would have to overcome this hurdle, possibly through hormonal therapy or by carefully selecting the largest and strongest lionesses as surrogates. Let's assume scientists manage to overcome every obstacle, and the world's first cloned Smilodon kitten is finally born. But that's where its problems really begin. A living organism is much more than just its genes. It also includes the trillions of microorganisms living in its gut, what we call the microbiome. These bacteria are crucial. They help digest food, shape the immune system, and significantly influence overall health. The Smilodon kitten's microbiome will be inherited from its surrogate lioness mother and shaped by modern milk and food. Yet its own digestive system is genetically wired for an Ice Age diet and consequently a vastly different set of bacteria. This biological dissonance, this fundamental mismatch, could lead to chronic illness, severe indigestion, allergies, and a severely compromised immune system. We might resurrect the predator's physical body, but we can't recreate its internal microbial world, and that fact leaves it fundamentally vulnerable. This brings up another fundamental question. Will a cloned Smilodon actually act like one? A huge portion of animal behaviour isn't just hardwired instinct, it's learned. Kittens learn to hunt from their mother. She teaches them how to stalk prey and deliver the killing blow. Our Smilodon cub will be raised by a surrogate lioness. She will teach it the lion way of hunting, chasing down prey, and working within a pride structure. However, the Smilodon's entire physique, its powerful forelimbs and fragile fangs, was engineered for a completely different tactic, ambushing slow, large prey. It had to tackle the victim to the ground before ever deploying its signature fangs. If it tries to hunt like a modern lion, it risks severe injuries or even fracturing those priceless sabers. Ultimately, we might end up with a creature that is genetically a Smilodon, but behaviourally it's just a confused, clumsy lion. Now we shift our focus from technical challenges to the profound ethical questions. Do we even have the fundamental right to bring an extinct species back to life? Some scientists and philosophers argue that it is, in fact, our moral obligation. Humans caused the extinction of countless species, and if we possess the technology to correct those historic mistakes, then we are duty-bound to use it. This would be an act of restorative justice. Opponents, however, raise a critical counter-argument. Isn't this simply the height of human arrogance? We begin to play God, deciding which species deserve to live and which must remain in the past. We can't ask the Smilodon itself if it wants to return to a world that is entirely alien to it, a world stripped of its natural habitat, its specific prey, and its own kind. We make this decision unilaterally, driven only by our own scientific curiosity and ambition. Let's face it, the first Smilodon born, and every single one after it, won't be a wild animal. It will be a laboratory exhibit. Their entire lives will be spent under constant observation in sterile, contained enclosures. Every action they take will be documented, every meal meticulously analysed. They will simply be the subjects of endless, invasive research. We can't simply release them back into the wild. Doing so would be both irresponsible and cruel. But if we don't, we are creating a living creature condemned to perpetual confinement. Moreover, the process of cloning is far from a perfected science. We already know that many cloned animals suffer from debilitating birth defects, premature aging, and a whole host of chronic illnesses. Are we truly prepared to take responsibility for the inevitable suffering of a creature we brought back purely out of scientific curiosity? But let's play out the ultimate fantasy. The project works perfectly. Scientists have successfully created a healthy, self-sustaining population of saber-toothed tigers. What happens next? Where on earth do we put them? Do we release them into the African savannah? They would immediately compete with lions and leopards. preying on animals that have zero natural defences against such a formidable predator. This could utterly destroy the delicate balance of an entire ecosystem. Or do we try releasing them in North America, their ancestral home? Today, that land is filled with people, grazing cattle and hiking tourists. Introducing a 300-kilogram apex predator would lead to absolute catastrophe. The resurrection of one species risks putting dozens of currently living species in jeopardy. We risk creating an invasive species that would make the rabbit problem in Australia look like a minor inconvenience. In this context, it's really helpful to look at the Woolly Mammoth De-extinction project, notably the one spearheaded by companies like Colossal Biosciences. Why are they so confident in its success? And why is their project considered both more realistic and ethically sound? First, they possess a far greater amount of high-quality genetic material, which was recovered intact from the Siberian permafrost. Secondly, their chosen surrogate, the Asian elephant, is a much closer relative to the mammoth than, say, a lion is to a Smilodon. But the most crucial difference is that the Mammoth De-extinction project has a clearly defined ecological mission. Scientists argue that reintroducing these giants to the tundra could help restore ancient grassland ecosystems, slow the thawing of permafrost, and even serve as a tool to combat climate change. This gives their work a vital global significance that transcends simple scientific curiosity. This is precisely why the Mammoth Resurrection Project can be viewed as the ultimate icebreaker for the entire field of de-extinction science. If the colossal team manages to successfully engineer and deliver a viable mammoth calf using an elephant surrogate, it will represent a monumental breakthrough. They will, in effect, pioneer and perfect the technology of interspecies surrogate pregnancy for long-extinct animals. They will tackle issues like immune rejection, hormonal synchronization, and numerous other complex biological hurdles. All this accumulated knowledge and technological expertise can then be adapted for other, more complex de-extinction goals, including the resurrection of our own saber-toothed cat. A successful mammoth birth will yield invaluable practical experience and, critically, draw massive new funding into this nascent field. Therefore, as paradoxical as it may sound, the road to reviving the saber-toothed cat runs straight through the Siberian tundra and the existing herds of elephants. There are even more exotic ideas out there, ones that sound like pure science fiction, yet are being discussed quite seriously. For example, what if we stopped trying to clone a living sentient creature, thereby avoiding all the associated ethical pitfalls? Instead, what if we create a biological avatar? Using synthetic biology, we could grow the Smilodon's muscle tissue, bones and other organs, in vitro, and assemble them into a complex biorobot. This creature would be controlled by an advanced artificial intelligence, trained rigorously on all available paleontological data regarding the predator's behaviour. We would essentially have an exact replica of the Smilodon, allowing us to study its biomechanics and hunting instincts, all without creating a conscious mind that would suffer in captivity. While this is certainly a matter for the very distant future, such an approach would successfully circumvent many of the ethical traps inherent in traditional cloning. Finally, we have to address the elephant in the room: money. Bringing back the Smilodon would come with an astronomical price tag. We're talking hundreds of millions, if not billions, of dollars. That same money could essentially save almost every existing big cat species from extinction. Amur tigers, snow leopards, and Amur leopards, just to name a few. Every year, we lose dozens of species, and the resources dedicated to conservation are already stretched thin. This raises a crucial, pressing question. Is our obsession with de-extinction a dangerous distraction? Is it pulling vital attention and funding away from the real and immediate problem, saving our planet's biodiversity right here, right now? Why spend billions reviving a single extinct species when those same funds could save hundreds of living ones? It's a difficult ethical choice, and, unfortunately, there is no easy answer. So, when exactly can we buy a ticket to Pleistocene Park? Let's get real. Despite all the hype and bold announcements, we are still at the very beginning of this process. Even the most optimistic forecasts suggest the birth of the first baby mammoth is still at least 5 to 10 years away. The Smilodon project is exponentially more complicated, primarily due to DNA degradation issues and the total lack of a close living relative. Leading geneticists largely agree that if this ever happens, it won't be for several decades, maybe even a century from now. Science still needs to solve dozens of fundamental problems before that can happen. So, don't hold your breath, waiting for saber-toothed cats to appear in zoos anytime soon. Right now, this remains more of a powerful scientific dream, a massive stimulus for technological advancement, than an actual engineering project with concrete deadlines. Ultimately, the true value of de-extinction projects might not be bringing back the saber-toothed tiger or the woolly mammoth itself. Their real significance lies in the underlying technologies developed along the way: gene editing tools, methods for handling ancient DNA, and in vitro fertilization techniques. All of these can and must be applied immediately to save species currently teetering on the brink of extinction. For instance, geneticists are already using these methods to boost genetic diversity in black-footed ferrets, or to try and save the northern white rhino, of which only 2 females remain. Technologies created to resurrect the dead could very well be the last chance for the living, and perhaps that ultimately is the greatest legacy of this ambitious de-extinction effort. All right, let's summarize where we stand. The journey toward bringing back the saber-tooth is a maze packed with almost insurmountable roadblocks. Chief among these are securing viable, high-quality DNA, the massive hurdle of finding an appropriate surrogate, and the overwhelming ethical burden we'd have to shoulder. While this might be theoretically feasible far down the road, making it happen in reality is an astronomically complex undertaking. Odds are, the woolly mammoth will be the very first animal we successfully bring back, and its ultimate success or failure will dictate the future of all de-extinction projects for decades to come. So, the return of the Smilodon remains a beautiful, yet currently unreachable dream. But the mere pursuit of this goal is already transforming our understanding of science, forcing us to peer into life's deepest mysteries and confront uncomfortable questions about our responsibility on this planet. Thank you so much for sticking with the video until the end. If you enjoyed this journey, please make sure you hit that like button and subscribe for more content. Now for the big question I have for all of you. If you could bring back just one extinct creature, who would it be? Drop your answers in the comments below. I'm really looking forward to reading your choices.